Welcome back. In the last lecture, I described how DNA is the gene and protein is the phenotype, and genetic arguments were used to establish this relationship. I want to continue that line of reasoning in this lecture and also describe how the phenotype plays itself out in the overall chemistry of tissues. My opening story concerns a human genetic disease. The Norwegian mother watched her 6-year-old daughter and 4-year-old son get progressively sicker over time. It was mid-1934, and both children were now profoundly, severely mentally retarded. Her family physician told her he couldn't help anymore and advised her to see a Dr. Asbjørn Folling, who is a medical specialist in both chemistry and mental retardation. Now, Folling saw the children with the mother and figured that the children's symptoms, this mental retardation and some other symptoms, might be related to blood sugar. Now, sugar contains a chemical grouping called a ketone. It's carbon with some oxygens present. We don't have to know the chemistry as such, but I just want you to know that sugar does contain ketones. So Folling decided to test for excess ketones and an abnormality in sugars by testing the urine and adding to it a solution of a chemical called ferric chloride. Ferric chloride happens to be brown in color. I've used it myself in the lab. And when you add it to ketones, the solution turns purple. And that's a diagnostic test of the presence of ketones derived from sugar. So Folling did this. He added some ferric chloride to the urine of these two children, and lo and behold, the urine turned green. Not purple, green. And he said, I had never seen this before. What's going on? There's a ketone there, but it's doing something different to the ferric chloride and turning it green. And what is this stuff? Well, he first had to rule out that whatever was causing this color change came from the children's own body chemistry and not something they ate on the outside. So he did an experiment. His hypothesis was that this green color changing stuff came from something the kids were taking. Maybe one of their special medicines. They were on a whole bunch of medications that the mother gave them that the other doctors had prescribed. And so his hypothesis was, well, those medicines have something ketones in it that are causing this green color change. That these ketones have nothing to do with the inherent biochemistry of these two children. So to test the hypothesis, he asked the mother to stop giving these kids any of the special medications they were taking for the next week. This would clear them out of their system, and then he retested. The result was the green color was still there. Now, that means he could reject this hypothesis that the green-colored substance, the substance that turned ferric chloride green, came from the outside, because when you eliminated the outside stuff, the green color was still there. So it must be from the body chemistry. I mentioned that Folling was a medical specialist in chemistry as well as mental retardation. So now he used his knowledge of chemistry. If you talk to your physician, you might ask them, how was pre-medical education? And very often they will say, well, the toughest course was chemistry, organic chemistry, they call it, the chemistry of carbons. That always has the reputation of being a tough course for students who are headed for a medical career. Well, Folling actually did pretty well in chemistry and did doctoral level work in it. So what he did was extracted this mystery substance, whatever it was, causing the green color, from over a period of many, many long hours in the lab. He extracted this substance from the urine of these two children. And he identified it. And the identity was something no one had ever seen before in urine, and it was called phenylpyruvic acid. That's the name. I'm going to give it to you for a reason. Phenylpyruvic acid. It has a ketone in it, and so that's what caused the color change in ferric chloride from brown, but not to purple, to green. Falling now named this disorder in his 2 children. He looked in the literature and scientific journals. He'd never seen anything like this described with this strange chemical imbalance. And he gave it the rather cumbersome Latin name imbecilitas phenylpyruvica. Sounds descriptive. Imbecile was an old name. We no longer use it to describe a severely retarded person. Phenylpyruvica is, of course, phenylpyruvic acid. Later on, it was called phenylketonuria, the ketones from the phenyl. The phenyl is a chemical grouping. So phenylketonuria in the urine. So I will abbreviate phenylketonuria, PKU, phenylketonuria. Over the next decade, Falling saw other mentally retarded people who put into their urine this phenylpyruvic acid, so this combination, and he made the diagnosis of PKU. Over half of his PKU patients were siblings, brother, sister, brother, brother, just like the original pair of patients. And in all of the families he saw when he had siblings, both parents were mentally normal and they did not put this unusual chemical, phenylpyruvic acid, into their urine. Now this is the 1930s, 1940s. Falling knew Mendelian genetics, and he realized that PKU must be inherited as a recessively inherited allele. Because both parents were carriers, they had a normal allele taking care of their phenylketones, not allowing them to be put into the urine, whatever the chemical error was, and they also had the bad allele that would cause PKU. The chances of having a child with PKU, therefore, are 1/2 of the gametes the mother is going to make will have this bad allele, 1/2 of the gametes that the sperm that the father makes will have the bad allele. So the combined probabilities is 1 in 4. And when he had large families, typically it was 1 in 4 children who had PKU. Now you might say, well, how did it happen that this woman was bringing in 2 children who had PKU? She must have had 8 kids, right? And the other 6 were normal. That's a lesson in statistics. In the average, it's going to be 1 in 4, but it's possible, certainly possible, to have 2 consecutive children who have this 1 in 4 chance of inheriting the recessive allele. Other human genetic diseases were known by Falling's time. In 1886, into the 20th century, an English physician named Archibald Garrod began to see patients with a rare disorder. The rare disorder was called alkaptonuria. Again, it's something in the urine, but in this case, the urine turned black when it was exposed to air. Garrod noted that this disease occurred most often in children of first cousin marriages. You know, first cousins have a grandparent in common, and he concluded that this must be a genetic disease because the first cousin marriages seems to be more common, and it's caused by a recessive allele. Garrod took this one important step further in studying this particular disease. Enzymes had just been discovered. In fact, the idea of enzymes was first discovered literally at the turn of the 20th century, in the first couple of years of the 20th century. And Garrod, I told you, was working at the end of the 19th and the early 20th centuries. And it was a huge publicity of what enzymes were, their catalysts and their proteins. Garrod proposed, he went on a limb, and he proposed that the error in alkaptonuria was expressed as a lack of an enzyme activity, the lack of a functional enzyme, and that the normal function of this enzyme is to convert some product of protein breakdown. He didn't know what it was. Knowing about genes and proteins, he came up with the one gene, one protein idea, long before the people who worked on bacteria did, as I described in the previous lecture. Garrod called alkaptonuria an inborn error of biochemistry, and to this day, there are journals of inborn errors, and there's a society of inborn errors. People still use that term. Inborn, of course, means genetic. And Garrett made the following statement, quote, "I believe that no two individuals are exactly alike chemically any more than structurally." Let me repeat. I believe that no two individuals are exactly alike chemically any more than structurally. An amazing and true prediction. We are all unique in our genetic makeup and in our phenotype, as it turns out. And Garrett said this 100 years ago. I want to talk about protein as the phenotype now, and biochemistry as expression of the phenotype. A biochemical pathway, as I described in the last lecture, describes a sequential conversion of substances in the body. Each step in a biochemical pathway, as I said in the last lecture, is catalyzed by its own specific protein, its enzyme that is encoded for by a gene. So we can envision a gene, DNA, encoding or having the information that will determine an enzyme. The enzyme folds into a three-dimensional structure because it is a protein that can do that. And you recall, proteins do this in a spontaneous fashion, depending on the order and chemistry of the amino acids in that order. And these enzymes then bind substances, line them up to allow products to form. The substances that bind to enzymes are called substrates. A substrate is a substance that is acted upon by an enzyme to form a product. And the products, well, they're called products. Now back to a biochemical pathway. A single biochemical pathway involves the enzymes that are deficient or that have inherited errors in their genes in a wide variety of diseases, including the two that we have described. In phenylketonuria, PKU, in alkaptonuria, in a couple of other diseases. For example, in albinism, albinos lack a product that comes from the product of phenylalanine metabolism. So phenylalanine is converted by an enzyme into its product, and that ends up being converted into pigment. And if the enzyme that converts the product into pigment isn't there or isn't active, you get albinism. So several genetic disorders exist in this and every biochemical pathway. Now, some pathways are more important than others, and so there are some genetic disorders we never see because the people just don't get born. Imagine a genetic disorder in the ability to have cells have energy in some way. If you had that, you couldn't have any energy. The cell couldn't do anything, and so it's not going to survive much in the embryo. Phenylalanine, which is the substrate involved with PKU, gets converted into this phenylpyruvic acid because phenylalanine, it's a side shunt pathway that normally doesn't happen. Phenylalanine normally gets converted into another amino acid in the liver called tyrosine. Now, phenylalanine is an amino acid, and so it's one of the essential amino acids we cannot make. And so we take it in our food. Now, when proteins containing phenylalanine are taken in, like for example corn protein, they are digested in the intestine to their individual building blocks, the amino acids. And one of those amino acids happens to be phenylalanine. Now here's what happens in the body after digestion. The amino acids, or any digestion products, end up in the blood. The blood goes from the intestine, and the first stop before it goes to the heart or anywhere else in the body is the liver. The liver is an amazing organ. It is what I call a chemostat. You know, a thermostat is a machine, piece of machinery that detects the temperature in the atmosphere surrounding it and says, oh, it's going to, and then sends an instruction, turn on the heat or turn on the air conditioning, depending on the temperature. That's a thermostat. So it keeps the temperature at the level that you want. The liver is like that. It's a chemostat. It looks at the chemical composition of blood that arrives in it and then says, well, should we convert some of the chemicals that arrive into other things There might not be enough blood sugars, so we'll convert some of the protein into sugars, etc. All those conversions go on in the liver. Now, in the liver, typically, some of the phenylalanine that is part of proteins is converted to other substances, and each of these conversions is catalyzed by an enzyme present in the liver. The enzyme that is responsible for catalyzing the conversion of phenylalanine into its product, which is another amino acid called tyrosine, has a name. It's called phenylalanine hydroxylase. That's the name of the enzyme that can do this. And the product, as I mentioned, is tyrosine, which is phenylalanine with a hydroxyl group. That's why it's called hydroxylase. To summarize, what are we dealing with in biochemistry here? The substrate of this reaction is phenylalanine. The product of this reaction is tyrosine. The enzyme that speeds it up is called phenylalanine hydroxylase. And it's the enzyme whose structure and function are determined by DNA, the gene. And I mentioned, of course, the other genetic disorders along this pathway. Now, each enzyme in a pathway is encoded for by its own gene, and genes can be changed in some way. That is, the information content of DNA can be changed. We will specify this later on in the course. And by changing the information content in DNA, you can change the expression of that information content as protein. And if you change the order of amino acids in protein, the expression, the protein will fold differently. And if it doesn't fold in the right way, it will not present the surface for the substrate for phenylalanine to bind and do its thing and get converted into tyrosine. The parents of the children who had PKU that Falling originally saw were not mentally retarded, as I mentioned. They didn't put phenylpyruvic acid in their urine. Why? Because they had one of their two copies of DNA coding for this enzyme as being the good copy, the DNA that had not been changed, the DNA that had not been mutated. They were essentially carriers of the mutant gene, the mutant allele. I want now to turn to the overall aspects of biochemistry and how that reflects the genetics of an organism. Metabolism is a term that you often hear, and it is essentially the sum total of all of the chemical transformations that occur in some sort of biological entity. It could be the metabolism of a cell, a single cell's metabolism. It could be the metabolism of the tissue, a muscle. So we can talk about muscle metabolism. That means all the chemical reactions and changes going on in a muscle. Or it could be the metabolism of a whole person, a whole organism. So we talk about the metabolic rate of a human's metabolism. Some people are high metabolizers, others are lower metabolizers. That's a general term for saying they just do a lot of chemistry in a high level or low level, especially in regards to energy metabolism. Metabolism is the expression of the phenotype, and much of the metabolism involves energy, transferring energy. I mentioned earlier on in the course that there are three major ideas that tie biology together. Remember, mechanism, cell theory, evolution. Mechanism of metabolism is reflected by the study called thermodynamics. Thermodynamics is the physical study of energy. Thermo, heat, dynamic, change, changing heat. Well, it's not really heat, it's energy. There are lots of forms of energy. There's heat energy, there's chemical energy, there's light energy, you know those three. And there's electrical energy, there's nuclear energy. There are two laws of thermodynamics that physicists, chemists, and biologists agree apply in biological systems as well as the rest of the universe. The two laws are appropriately called the first law of thermodynamics and the second law of thermodynamics. Really good names. The first law says that energy is neither created nor destroyed. If you look in the whole universe, the sum total of the energy is constant. All we're doing is changing energy into one form or another. We can't create energy. This means that in living tissues, in any metabolic conversion, we're just changing energy from one form to another. Let me give you an example. We eat some sugar, good food energy in sugar. And the sugar gets converted ultimately into carbon dioxide, which we breathe out. So our reaction is sugar, lots of bonds, and sugar and carbon dioxide. Now, sugar has a lot of energy stored in its chemical bonds. I'll give you the formula for sugar. C6, remember H2O, C6H12O6. Lots of carbons and oxygens and hydrogens all tied together. And they're tied together, the tying together requires and stores energy. CO2, carbon dioxide, not much energy at all. Well, where's the energy go? We're converting this complex thing that has a lot of stored energy into a simple molecule that has very little energy. Energy is neither created nor destroyed. Some of the energy goes off as heat, and that heats up our body, by the way, and other aspects of this energy are used immediately by reactions in the body that need energy, like, for instance, making fat. So if you eat excess food of carbohydrate, you can use that excess energy. Well, you don't really take it off as so much heat. The body doesn't want a lot of heat coming off. So instead, you use that energy chemically to store it as fat. That's the first law. The second law of thermodynamics says that in any conversion involving energy, disorder increases and usable energy is lost. I said, some energy comes off as heat. Well, if we trapped all that heat and we trapped all that chemical energy, we're still missing some energy. Chemists and physicists recognize that the universe is kind of running down, that the tendency of the universe is to have energy in a form that is disorder. We call that entropy. There's an old Woody Allen film that I recall in which Woody has a kind of daydream in which he recalls his youth, and he's a young kid in the 1940s, and his mother has taken him to the doctor because Woody is very depressed. He just read in a magazine that the sun, all the chemical reactions going on in the sun, are converting one energy into another, and some of it's going off as heat, and some of it's going off as light, but some of it is being converted to this unusable form, entropy, and gradually, astronomers have said in the article Woody has read, the sun is running down and will be out of usable energy, and the Earth will no longer have life on it at that point because there won't be any more sun. The kindly doctor, who is smoking a cigarette, as I recall in this scene, says to Woody in a kindly way, but that's not going to happen for billions of years. And Mother, of course, screams at Woody, saying, here you at billions of years, go do your homework. Stop worrying about it. Now, what this idea of unusable energy in biology means is that it requires an input of energy to make complex substances from simple ones. If you want to build a complex protein, you're going to need energy for that building, and you'll see how this happens later on in the course. It's just like a teenager and cleaning up his or her room, requires an input of energy to do so. So, mechanism tells us biology is part of the physical universe, and life obeys these laws of thermodynamics, just like the sun does. We recognize in terms of energy two types of chemical pathways in cells and tissues, in living tissues. These two pathways are called catabolism and anabolism. Catabolism means breaking down complex substances like sugars to simple ones like carbon dioxide and releasing energy for use in the cell. So that's what catabolism is, breaking down the complex to the simple and releasing energy. Example, of course, being the breakdown of sugar. Anabolism, anabolic things build up. So anabolism builds up complex substances and therefore requires energy. So anabolic steroids are drugs or hormones that cause a buildup of muscle tissues. And you can get all buff, etc. I always tell my students that I'm doing that in the gym every day, and they laugh because I'm a rather thin person. There are several rules for metabolism that biological tissues obey and that are genetically determined. And these two rules are, first, metabolism occurs in small steps to release energy in small usable amounts. And secondly, that metabolism and conversions and pathways are genetically determined. First, the small steps. Consider sugar, as I mentioned before, in catabolism, the breakdown, being converted into carbon dioxide, this simple molecule. I mentioned releases a lot of energy. Those stored bonds in the C6H12O6, all these bonds between the carbons and the hydrogen and the oxygen, that stores a lot of energy. The amount of energy, if you did this in one fell swoop, one single reaction, is 50 times more than a cell would ever need for a single anabolic reaction in the cell, a single building up chemical reaction. So you'd lose a lot of the energy. You'd lose 98% of it because you don't need that much energy. It's like using an atomic bomb for a firecracker, right? So this is not really good. You're going to lose most of the energy as heat. Instead, in biochemistry, in this cell, this conversion of C6H12O6, of this complicated sugar, into simple CO2, carbon dioxide, happens in 40 steps. And the energy is released bit by bit. And each bit then can be used efficiently for anabolism. There's about a dozen steps in this conversion in 40 steps that actually release some energy, because you have to rearrange the molecule. It's a fascinating series of events that biochemistry students have to learn. Because each of these conversions is catalyzed by an enzyme and therefore determined by a gene, the pathway, the overall pathway I've just described, these 40 events that happen going on to CO2, is genetically determined, because there's a gene for an enzyme for each one of the steps in this particular pathway. Just as I could say A to B to C to D, there are 40 steps involved. Let me give you an example of genes determining biochemical pathways, because there's a great diversity in nature. Just getting energy out of sugar, well, a lot of organisms can do that. Let's look at some that are particular. I'll talk about the lowly bacteria. They're not necessarily lowly, they're just complicated in their own way. For instance, bacteria have genes that determine the enzymes that can take carbon, hydrogen, and oxygen and mix them up and put them together in a simple sugar to rearrange them to make ascorbic acid, also known as vitamin C. We can't do that. We can't rearrange atoms to make vitamin C. We don't have that gene and that enzyme. Another example, there's some other bacteria that can take cellulose, which is paper or indigestible things that we can't digest, from wood or paper or grass, and convert this cellulose, which is a polymer of glucose is hooked together, and convert it into glucose sugar. Glucose is one of the sugars. And so they can use this sugar for energy. We can't do it. We lack the gene and therefore the enzyme for this biochemical pathway. Well, of course, there are many things that we can do and that bacteria can't. So, we can get back at them in our own way. My point is, metabolism, sum total chemical reactions, means phenotype. And this is determined ultimately by genes and the enzymes that they determine. We now turn in the next lecture to the way that DNA as the gene actually determines protein as the phenotype. Thank you.